Hi everyone, my name is Tejal Nair and I am a summer trainee in the Mayan lab at the Icahn School of Medicine at Mount Sinai. Today I'll be presenting on gene gauging, a web interface that displays how the expression of genes changes with age in different human tissues. So first, let's begin with some background and motivation for the project. Aging is a multifactorial process that is influenced by interactions between the environment and the genome. The diagram on the right categorizes the hallmarks of aging into three groups. These hallmarks are the biological and chemical changes that occur inside cells, tissues, and organs as we age. Aging research has identified several key genes that play critical roles in aging and age-related diseases. So using the GeneShot search engine, a resource developed by the Mayan lab, we identified the top 10 genes that are co-mentioned with the term aging. The table displays these genes along with their total publication counts with aging and the proportion of publications that co-mentioned aging. A critical component of the project is ArchS4. ArchS4 is another resource that was developed by the Mayan lab. It provides access to uniformly aligned gene counts and transcript counts for all human and mouse RNA-seq datasets from the NCBI Gene Expression Omnibus. Led by Dr. Alex Lockman, a research assistant professor in the Mayan lab, ArchS4 currently contains over 1 million samples. For around 89,000 of these samples, the age of the patient is available in the metadata. Additionally, we identified 16 tissue types with over 150 samples that have this age annotation. Another important resource for the project is GTEx. This NIH Common Fund program's primary goal is to profile the genomics and transcriptomics in human tissues. It provides gene expression data collected from over 900 postmortem donors across 54 different tissues. The expression profiles have limited publicly available metadata, but it does include the age of each sample in 10-year bins. So for our analysis, we consider the age of a sample as the median within each age bin to maintain consistency with the ArchS4 dataset. Next, using the ArchS4 Python package ArchS4Py, we were able to download the relevant samples from ArchS4, and we also download the data directly from GTEx. We focused on 16 major tissue types. The CSV file in the upper right corner contains the listings of the samples for each tissue. This file was used for organizing and pre-processing the data for analysis. Additionally, gene expression data was normalized using the ArchS4Py normalization function. Using those samples, we plotted the gene expression data against age for each human gene. The next step in the development of gene gauging was to set up the user interface. So there were three main components we focused on while developing this. These include dynamic content loading, plot visualization, and usability. To enhance user experience, we incorporated a search bar for gene names. Also, once a gene is selected or typed in, we display its similar genes and transcripts based on those that most correlate with the selected gene. By including interactive filtering options, users can focus on specific plots, and these plots can be viewed in a larger pop-up window. There is also an option to download the plots and the data used to create the plots. Now I'd like to quickly demo the site. As you can see, there are 4 input areas on the left side of the screen for 3 different filtering categories. These include gene, tissue, and resource type. And when a user selects a gene, they also have the option to select a transcript type, which you can see in the center of the screen. These filters determine which plots will be displayed. And here's the download section with all the data. So besides developing the web interface, we also did some analysis on the process and organized data. Here are the top 30 most positively and negatively correlated genes with age analyzed in the 16 different tissue types in the ArchS4 dataset. The histogram in the middle shows the distribution of Pearson's correlation coefficients for all genes with age. All genes PCCs are compared to the PCCs for aging identified using the GeneShot tool. This highlights that some known aging genes expression highly correlates with age, but most of them do not. Next, here are the top 30 positively and negatively correlated genes in blood tissue specifically. The results show clear upregulation of genes associated with inflammation, such as the IL-1 receptor and TTN, and the downregulation of keratins and FAM family genes. An interesting result is the downregulation of EPO, which is a well-known hormone that the kidneys naturally make to stimulate the production of red blood cells. Next, we aim to examine if the top positively and negatively correlated genes in blood are well studied and already known to be related to aging. While some of these genes are well studied in the context of aging, there are several novel understudied genes in both lists, as shown by the publication counts. For example, FBX030, which has only 20 publications in PubMed, is shown to be upregulated in the aging human blood. FBX030 and other similar genes with low publication counts are good candidates for experimental validation in the context of aging research. So, for example, it would be interesting to explore whether the gene products of these genes also correlate with age and whether knocking out these genes in animal models impacts the aging process. To conclude, let's summarize and outline some future steps. Gene Gauging is an online database with a user interface that provides plots about the relationship between aging and gene expression for single human genes across 16 different key tissues. By integrating data from ArchES4 and GTEx, users can observe how gene expression varies across different tissues as we age. To further enhance the functionality and usability of Gene Gauging, we hope to implement some new features. For example, users will be able to input their own single-cell and bulk RNA-seq datasets, and we should be able to predict the age of these samples based on the data sorted in Gene Gauging. We also plan to add data from the Clinical Proteomic Tumor Analysis Consortium, or CPTAC, proteomics and transcriptomics to Gene Gauging. CPTAC has gene and protein expression from normal tissues collected from over 1,000 cancer patients. So integrating proteomics data will provide insights about how gene expression is correlated with protein expression as we age. We also plan to add support for mouse data. This will enable us to learn how gene expression correlates with age in multiple tissues across different mammalian organisms. Finally, I would first like to express my gratitude to Dr. Mayan and Ms. Jenkins for this opportunity, as well as Alex, Nash, and Anna for their support and collaboration on this project. I would also like to thank all members of the lab, as well as my fellow summer trainees. Thank you.